Hi guys, happy Tuesday. I'm sorry I can't be there. I hope to be back later on in the week. It's my own kids' close contact situation, crash school, classes at home, that kind of thing. So just a sign of the times. But I hope to be back, and my apologies that I can't be there. But anyway, we're going to carry on. So I got you messing around with microscopes the last day, and you were trying to zoom in and zoom out and magnify and see your own handwriting and all that. And you were, what else did you look at when you're looking at the microscopes? You were looking at marker on paper, and you were looking at the top of your pen, and that's all well and good fun. But the idea of a microscope, basically for scientists, for people like us, is to see stuff that's small, usually things like blood samples and urine samples and even smaller things, the things that make humans and make animals and make plants, and I suppose mainly things like the cells. So I will show you cells. I will show you onion cells and I will show you human cells under the microscope when I'm back in the lab. But I suppose for now we can work on the theory and we can get the stuff that you'll need for your exams down in your hardback. Even though we can't actually do microscopes today. Okay, so this is just what you're going to learn. So what we're going to be looking at is things like this, things like so these are basically cells. Obviously not these ones here; they're bricks on a wall, but these ones down here are cells. They kind of look like bricks on a wall, but they are the building blocks of life. They are a load of cells in a row, piled on top of each other, and they're onion cells. Okay, so they look very similar to bricks on a wall. So they're very, very tiny, but they have all the characteristics of life. Another thing we have to look at. The smallest living thing that can exist is a single cell by itself. And what you are is a load of these cells combined together, and there's loads of different types of cells, and they all work in different ways in order to make something as complicated and as special that is a human being, that is something like yourself. Okay, so we have here is a single cell. So here's our single cell like that. And that cell can multiply and multiply and multiply. So if yours, say if you're walking down the hill from school there and you fall and you cut yourself, and what happens is you get a break between your cells. That's a cut in between two cells. What will happen is that this cell here will try and bridge the gap to this cell here, and what it will do is it will make new cells. And that's why if you cut yourself, it's not a permanent thing. The new cells will form around that. But the idea of what we're doing at the moment, the problem with cells is that they're really small. They're very, very small. I can't look at you and say, oh, look, there's your cells, because they're too small. So what we do is we use this thing, the yoke you were using the last day in the lab, we use a microscope. And a microscope basically allows us to see small stuff that we can't see with our eyes. Basically, our job today is to look at what's a cell made of. And bear in mind now, you have you're now a member of the OneNotes. You can go into your OneNote and you can look at these notes, and you can you can use them again and come back to them then when an exam comes up and so on. So, a couple of things that are in cells. In all cells, whether it be cells of a polar bear or cells of a penguin or cells of a cat or a snake or cells of a human being, all animals are just listed there. They all have three things, and three things only really that you need to know about. They have a membrane. Membrane is the bit around the outside. So your membrane is like the skin on the outside of a cell. It's your cell membrane. They have a nucleus, which is the blue bit in the middle there. And the red part then is cytoplasm, the liquidy part. So I kind of go into, so there's your three labels of all cells, but mainly of animal cells, because plant cells have a few more, which we'll see in a second. Each cell is like a factory. It has lots and lots of things that it does. So the cell has a one big job, but each part inside the cell has loads of different jobs. The first thing we're going to look at inside the cell is the blue bit in the middle, the nucleus. And the nucleus is the brain. Now, I don't like you thinking like cells think. They don't. But the nucleus controls all the activities of the cell. So it's the most important part of the cell. It contains all these, you've heard the word genes. You get genes from mum and you get genes from dad. They're passed down to you. Well, all your genes from mum and dad are contained in the nucleus in every single cell in your body. The membrane, the outside part, well, the outside part, it's like the outside part of a bubble. It's a thin layer surrounding the cell, and it decides what's allowed in, and it decides what's allowed out. So it lets stuff into the cell, and it lets stuff out of the cell. So it's like the walls in a prison. It decides what gets in and decide what gets out. Now, I know I'm flying through this, so we can go through this a bit slower now in person. And the cytoplasm, the red bit in the cell, is it's a liquidy stuff, and that basically gives structure. So if that wasn't there, it'd be all shriveled. So because you have all this red liquidy stuff all around the outside, it gives it that nice bubbly shape. If that wasn't there, it would just be like a squash tea bag, really. So it gives it structure, and it you get some sort of reactions that go on in there, which we don't actually do in junior cert. We'll worry about when we're in fifth year. Here's your three labels: your nucleus, which is your control center; your cell membrane, which is your bit that allows everything in and out; and your cytoplasm, which is your liquidy bit. So there's an animal cell with three labels, and here's a real microscope image. What I hope that you'll be able to see when you swab your own cells. And that's where we get the animal cells from. We get it from you. And there's an animal cell. If we zoom in on this one here, our membrane's on the outside, our nucleus is the dark spot in the middle, and our cytoplasm then is all the bits around the outside of the cell. Okay, this is going to be important. Remember this slide. We have a plant cell and we have an animal cell. So I've spoken about three labels so far: the membrane, cell membrane, the nucleus, the control center, and the cytoplasm, the liquidy stuff. Animal cells are quite basic, like a fried egg, really. And it only has three labels that do three specific things. If you're a plant, though, which most of you aren't, we have a look and we have extra labels on the left there. So we have the cytoplasm like the animal cell, we have the nucleus like the animal cell, and we have the membrane like the animal cell. But then we have three more labels. The plant cell has a wall around the outside, a vacuole, this big white space in the middle, and it has these kind of green M&M-y looking things all around there called chloroplasts. So there's a bit more to a plant cell than an animal cell. And there's a reason for that, which we'll get to in a second, right? So there's your plant cell there. The differences. So you saw that when you looked at the diagram, really. The animal cell was kind of a fried eggy, irregular, weird looking shape, whereas your plant cell was like a block in a wall, or a square, or a rectangle. The plant cell has a cell wall, it has chloroplasts, and it has a vacuole. So they were the three extra labels that the animal cell didn't have: number 1, number 2, number 3. And we saw then this is just the opposite, that the animal cell has no cell wall, no chloroplasts, and a couple of really, really small vacuoles that we don't even see. So you could even say, get rid of that there, and it has no vacuoles. You'd be safe enough saying that, because it's not something that we can see on a microscope. I just put in a thing there for you to that you can fill in, basically plant cells. So all the stuff that are in plant cells go here, all the stuff that are in animal cells go here, and all the stuff that are in both of them go in the middle. So if you've started sets of maths, and this is a Venn diagram of the commonalities between animal cells and plant cells, and I've put it in a nice table there for you to take a look at and take a read at. Okay, the other thing I want to do today, now there's a few other bits there that I'm skipping over, but there's another thing that I want to go through today, and it's down here, is the microscope. So if we go down, I'm just going to flicking down, and this part here. So there's a bit more that I'm actually going into today, but this is a microscope. This is the thing you were messing with in the senior lab last week. And the microscope, you have a couple of important parts that you were using the last day, but you probably didn't know what the names of them were. And I'll show you. I'm just going to show you really quickly what they are now. So the bit you were looking into was the eyepiece. And there's a little lens inside the eyepiece that kind of magnifies whatever you're looking at. Remember you were saying to me, how do I carry this without breaking it? And this part down the back here is just called the arm. And if you're carrying a microscope, you grab it by the arm. You're not going to break it if you're carrying it by the arm. These lenses, remember you were turning to get the low power and the high power? They're called objective lenses. So you have two lenses in a microscope. You have the eyepiece lens up the top, and you have the objective lenses down here. Now, you know, even in the objective lens, you have three of them. You have 1, 2, 3. So you have 3 objective lenses because you have low, medium, and high power. The thing you were putting your pen on and trying to clamp it in place was called a stage. You have 2 knobs on the side. You had the big one here, and you have a small one. You asked me what do they do? And they're for focusing. So you were, remember I was saying to you, you put the stage to the bottom, and you look in, and you keep turning it back and back and back until it comes into focus. And what you were doing was you were turning the coarse focus knob and the fine focus knob to bring it into focus. The things that held your pen in place were just called clamps or clips. And before you started all this, down the bottom of the microscope, you had the light. So you have to turn the light on. Because when you turn the light on, the light shines up and up and up into your lens, up through and into your eye. And that's what allows you to see. And the reason it was difficult to see your pen was because the light couldn't travel through the pen. That's your microscope. And down here, I've put a table in for you of what each part does. So we kind of spoke about it there in the video. You can rewatch this video. It's on YouTube. We have the function of the eyepiece, the focus wheels, the objective lenses, a stage, a slide, and the light bulb, and what they do. So they usually either magnify, or they focus, or they hold the slide, or they supply light. They're your main functions of the microscope. Okay, so there's 2 things then I want you to focus on today, and you're going to be working in your hardback for this. And I'm just going to flick up to where I put in to tasks, and bear in mind you all have access to this document. There's a massive gap between one set and the other set, but sure, I can fix that before you get a chance to go into it. So what I want you guys to do today for the rest of this lesson and for your homework for the next day is this. In your hardbacks, I want you to draw a microscope. You can look one up, similar to the one we've done, or you can use this OneNote. You can go back down to those slides and draw it from that. If you happen to be in a lab and the teacher allows you to take a microscope and put it on your desk, you can draw it from that. If you're good at sketching, you may do that. Most importantly then, I want you to label your microscope. And under each label, so you're going to have the drawing, the label, and what's the function of each label. So on the one page, you're going to have the picture, the labels, and the functions. When you're finished with your microscope, I want you to draw a plant cell, and I want you to draw an animal cell. Again, you can use the OneNote. You can use the same notes are on Schoology, or the same notes are in your Exploring Science textbook from Edco, which is in the Edco app. That has the animal cell, plant cell, and the microscope as well. So whichever suits. Again, label the plant cell, label the animal cell, and for each label underneath, what does it do? So if you draw the animal cell and you write nucleus, write what the nucleus does. That's important. As I said, all info is in the notes above and below this instruction, so you can use you can log into OneNote and do that, or you can use your EDGO textbook, or you can use Schoology. All the notes are there for you, and we'll hopefully later on in the week we will get a chance to actually use the microscopes and see what each part of the microscope does, and actually see the cells. Can we actually see some animal cells and plant cells and how they work? Okay, thank you. See you later.